All right, everyone, thanks for coming out for Evan's thesis defense. We're very excited, always excited when we have a student get up and defend and do their culminating experience. So first I wanted to thank Evan's committee members. So Dr. Cheryl Logan from CSU Monterey Bay, and then Dr. Sue Sogard from the Southwest Fisheries Science Center up in Santa Cruz. Both of them were co-PIs on an NSF project to look at how ocean acidification and hypoxia affect the behavior, physiology, and gene expression in juvenile rockfish, of which Evan's thesis was one of the major components of that project. So I want to thank them. I wanted to thank, and he'll tell you all about that coming up soon. I want to thank the ichthyology lab for the nice spread and the food that we'll have for the celebration afterwards. And then we'll start with a little bit of an introduction just to set the stage and kind of tell you a bit about who Evan is. So a little bit about Evan's background. He grew up on the East Coast in New York. He grew up on a small island called Fire Island off of Long Island. He went to a very prestigious high school. Is it the Tabor Academy or the Tabor Academy? The Tabor Academy, yeah. And then he went to Pepperdine University for his undergrad. While he was there, he worked a bit with Dr. Karen Martin on some of her grunion studies. And then his main advisor was Lee Katz, where he did a lot of work working in some of the streams kind of in the Malibu Hills, studying some invasive crayfish and some of the interactions in those stream communities, and then came here to Moss Landing to work with us on some of this climate change work with rockfish. I look back at his statement of purpose, and he was one of the few students that he pretty much did exactly what he described as his kind of goals for coming to grad school, which was great. So, a little bit about Evan as a person. When I tend to think about Evan, I tend to think, he's the man, right? The myth, the legend, right? Multi-talented man, very impressive in many ways, but what you might not know is that he actually has a few interesting phobias out there. One of them is that he suffers from this debilitating disorder called pterophobia, which is an extreme and irrational fear of being on the ground. Most all the photos I got of him were something like this. So potentially by the end of his presentation, he's going to be up on the stage, he might be up here on the podium, who knows? But he's been seeing a therapist, and he's really working on it, so he's here. Okay, another one. He suffers from another disorder called ergophobia, an abnormal and persistent fear of work. Well, mainly when he's down in Mexico and near a beach, for example. But he doesn't suffer from relaxation phobia, luckily, which is actually a word. I was surprised. Okay. But no, actually, in reality, Evan worked very, very hard for his thesis on his project. I've always been incredibly impressed with many of his skills. And as I got to know Evan, I started to think, maybe he might be the next MacGyver based on many of his skills. And we're going to go through some evidence here and see if this is actually the case, right? I mean, he's got the looks, he's got the hair, right? His mind is the ultimate weapon there, right? And so let's see, does anything line up? Well, I basically found out that he can build and fix really anything, right? He was the research assistant on an NSF grant, and while doing that, he devised all sorts of crazy, cool contraptions to measure behavior and physiology. We'd read a paper and say, hey, can we build this thing? And he would go and do it. So he's going to tell you and show you many of those things that he did, and he spent all these hours in the lab working this very temperamental pH and oxygen regulation system. As you can see on the far right there, still he cannot keep his feet on the ground. He's always climbing on things. So, yep, checks out, total MacGyver, right? Let's see some other things. Well, yep, pretty limited tools. He can escape from sticky situations, yeah? I think pretty much that lines up. He's a MacGyver. Let's see, right? He's capable of piloting many different types of watercraft, sailboats, motorboats. I don't know if he can do that seaplane, but checks out, yep. MacGyver. Other types of watercraft as well, right? So lots of different modes of transportation. Let's see, what else lines up? Well, he can accomplish very complicated tasks when he's under pressure, especially when that pressure is the water on him and the amount of time that he can use. So studying grazing and parrotfish here, spent lots of time underwater. So yeah, you think I can't fix it with duct tape? Do you even know who I am? And in this case, clothespins and zip ties are our underwater duct tape. Right, so multi-talented, communicating, delivered lots of posters and oral presentations at meetings like the Western Society of Naturalists. He can communicate through music beautifully. Right? He can communicate to the fishes and put dinner on their plate. Right? Again, multi-talented. He's an author. Here's a paper that he published from some of the research he did as an undergraduate at Pepperdine, looking at these stream communities. Right? He's helped raise money for his project, awards, the CSU Coast Graduate Research Award, the MLL Wave Award, lots of service, right, for the institution. He was a teaching assistant in our marine ecology class. He was student body president for a year. Right? Mentored probably more than that undergraduate researchers on our projects. Right? Participated in the open house. So, yeah, I mean, you think you give him some paper clips and yarn, and right, he could save the world, right? But then, you know, you do spend more time around him, and you start to see, well, maybe there's another side to him as well. And so maybe some of the times he's actually channeling MacGruber here. So those of you who don't know MacGruber, right, he was a Saturday Night Live parody for a number of years, right? And MacGruber, you know, in that situation, the bomb's about to blow up, he wouldn't save you, right? It would just blow up. Things would go crazy. So I don't know. Does Evan ever have any MacGruber-like tendencies? Well, I think so. I don't know. Would you trust your life to this guy? Is it all going to come crashing down? Is he prepared for any emergency? Yeah, I don't know. Here, I don't even know what's happening here, but maybe some other new phobias. But what I think there's actually a mathematical formula here that we can say that really he might be a mix there. MacGyver plus MacGruber is Evan Mattiasen. All right, so lastly, the other special celebration that's happening today is actually also Evan's birthday. So, happy birthday, Evan. All right. So without further ado, let's have Evan come up and tell you what he did for his thesis.
Okay, cool. Thank you, Scott. That was awesome. All right, so today I'm going to be talking to you guys about hypoxia and the effect on behavior and physiology on juvenile rockfish. Before I get into the talk, just want to give you guys a little bit of an outline or a map of how this talk is going to progress. So I'll start by talking about some global trends in oceanic oxygen, talk about some sources of hypoxia and their impacts on fishes, and then I'll introduce you to my research objectives and my study species. From there, I'll transition into the next section of methods and results. This is composed of about 2 parts. The first part is I'm going to talk about some of the behavioral experiments that I looked at, 2 experiments in particular, and then I'll transition to the physiological experiments, and there's 3 experiments for physiology. And so this is kind of a combined section where I'm going to introduce each experiment to you, run you through some of the methods, present the results, and then tell you a little bit about what those results mean, and then I'll move on to the next experiment. So progress through all the experiments, and then at the end I will summarize some of the results, give you a little bit more insight on what those mean, and leave you with some closing thoughts. Also, there will be a little bit of a section title here up in the left-hand side, just so you can kind of keep track of what section we're in as we go. So this talk is focused around oxygen. Now, oxygen is fundamental to biological processes in the ocean, and its decline can lead to changes in productivity and diversity of our oceans. Since the Industrial Revolution, human activity has led to a number of changes, including increases in sea surface temperature. Here we have a figure showing a 100-year average of sea surface temperature with warm colors representing an increase, as well as an increase in atmospheric CO2, which has led to acidification of our oceans, and also an increase in nutrient input. Here we have a figure showing chlorophyll concentration around the world. Chlorophyll is used as a proxy for nutrient input. And so you can see the warmer colors, particularly around coastal areas, that's showing some high nutrient input areas. So together, these changes have led to steady declines in ocean oxygen levels in both open and coastal systems. And now the figure on the right, I know it's a lot, but what all these figures are showing, and they're long-term time series of oxygen concentration in global oceans, and what they're all showing is that there's this general decline in oxygen over time. If we look at the figure on the right, this is representative, again, of changes in oxygen concentration, with warm colors representing a decrease in oxygen saturation. And the unit here is square meters of moles of oxygen per square meter per decade. So we've seen about a 2% decrease in oxygen saturation since 1960, and it's predicted to decrease by another potentially 7% by the year 2100. So that's the global view. And now if we take a look a little bit closer here in California and the California current system, these same trends are persistent. At the top of this figure, we see some long-term trends again showing decreases in oxygen here in the Monterey Bay. And then below that is some trends in coastal oxygen conditions for Central California. And what they're showing are some consistent decreases in oxygen. Now, hypoxia is the phenomenon where dissolved oxygen in the water column decreases to a point where it begins to have some detrimental impacts on the organisms living in that system. The threshold for this is generally agreed to be around 2 milligrams of oxygen per liter, which is equivalent to about 20% oxygen saturation level. And so here on the right, we have a figure showing a global map where areas indicated by red dots are coastal areas that have been directly impacted by anthropogenic inputs, leading to decreases in oxygen saturation and causing some hypoxia zones. And then also on this figure is some open ocean systems highlighted in blue, showing some expanding oxygen minimum zones. And again, what that is just areas in the ocean at the lowest oxygen concentration, usually found at a depth below 200 meters. So there are two categories that hypoxia can fall into, those caused by natural phenomenon and those influenced by anthropogenic impacts. And these processes are not mutually exclusive, and they often work together to change oxygen concentration of various systems. Some natural sources of oxygen change in the open ocean include inputs like air-sea exchange or wave activity. Also, photosynthesis through primary producers like algae or phytoplankton can also add oxygen to the system. But now as these primary producers then die, they sink down into the water column, and they can be decomposed by microbes, and that decomposition has some really strong respiration associated with it, which can decrease the oxygen content. There's other respiration through animals like zooplankton, and also denitrification can change the oxygen concentration of open ocean systems. Now, another one that is both natural and also influenced by inputs is warming of surface waters. And what this does is it creates these strong stratified layers, which leads to a decrease in mixing between the upper water column and the deeper ocean mass. Now, that upper water column is typically higher in oxygen, and the deeper water mass relies on mixing between these two to become more oxygenated. So increase in sea surface temperatures and therefore increase in stratification can lead to some strong decreases in deep ocean systems, hypoxic zones, or expanding oxygen minimum zones. Now, a common example that is given as a direct anthropogenic input affecting coastal system oxygen is nutrient over-enrichment. And this is through the introduction of nutrients such as nitrogen or phosphorus, as well as organic matter, which can create an explosion of phytoplankton growth in the water column. And then that process repeats itself as the phytoplankton die and then fall through the water column and are decomposed by microbes. And there's really strong, again, microbial respiration, which can lead to hypoxia in coastal systems. And in extreme cases, even death of an ecosystem, as you see represented by that fish right there. So in addition to direct land inputs, those open ocean water masses that can decrease in oxygen can also directly input coastal systems through a process that we know as upwelling. Now today's a great example of upwelling. If you took a look outside, you saw some strong winds. And so winds just like that are what initiate upwelling. But before I get into that, so upwelling is a process caused by large-scale gradients in atmospheric pressure, usually a high-pressure system over the water mass and a low-pressure system over the land mass. And this difference between pressure systems create these strong alongshore winds, in our case, running along the coast from north to south. And these winds initiate a process known as Ekman transport. And Ekman transport is when ocean currents are moving perpendicular to the wind source. And what it does is it moves the surface waters that are in the coast and it transports them out offshore. And now something has to come in and replace those water masses. And what does is that deep ocean water mass that's low in oxygen and high in nutrients. And it introduces those nutrient-rich, low oxygen water into the photic zone, into the coastal area, where, again, that cycle of phytoplankton or algal bloom occurs and associated microbial respiration, reducing the oxygen in coastal systems even further. So upwelling can have some pretty strong fluctuation impacts on coastal oxygen systems. And here we have a long-term, well, not a 6-month time series showing oxygen fluctuation between November 2010 and April 2011 with data taken by the Monterey Bay Aquarium in black and Moss Landing Marine Labs in green. And what it shows us is there's some regular fluctuations going past this sublethal threshold, which is about 4.6 or about 45 to 50% oxygen saturation, and even reaching that hypoxia threshold of 2 milligrams of oxygen per liter. So I just want to stress that these conditions are tightly correlated with upwelling winds. And here we have a 3-panel figure at the top showing wind patterns with the blue dips representing those alongshore winds. And what we see is that when we have these upwelling winds, there's also some strong correlating decreases in pH and oxygen. And then when those winds relax, the conditions go back to ambient levels. And so I haven't talked much about that pH aspect yet, and that is an important one that'll tie in at the end, but for now I'm just going to focus on the oxygen exposure aspect of this process. So now these pressure systems that create winds which initiate upwelling are sensitive to climate variability, and in particular changes in temperature, so as land temperature increases at a faster rate due to climate change, the thermal gradient between land and sea also increases, which leads to an increase in these pressure systems. An increase in these pressure systems then leads to increase in these strong alongshore winds, which increase upwelling events. So here we have a figure showing just that. On the x-axis is the difference in land-sea temperatures, and as you move right, that's a greater difference between the two areas. And then on the y-axis is offshore Ekman transport, which represents upwelling. And so we can see that there's a really strong correlation between these two variables. So as wind and climate change progress, here we have a figure showing wind patterns over about a 20-year period with warmer colors representing an increase in wind stress. And so as climate change progresses and wind increases across the coast, we expect to see an increase in upwelling events. So now we've talked about some processes governing ocean oxygen systems and some sources of hypoxia. So given that, what I wanted to know is how are juvenile stage rockfish going to fare under these future oxygen conditions? So from previous studies, we know that there's some potential impacts that can be caused to fish after exposure to oxygen, and they include reduced abundance or diversity. Here we have a figure showing density of schooling and benthic rockfish before and after a hypoxia event, and what we see is a dramatic decrease in those densities after that event. It's also predicted that the growth rate and body size of fish is going to dramatically decrease, and here's a model showing the predicted decrease across some of the oceans. And what they're generally showing is about a 10% decrease in body size. Now, some other physiological impacts that are known are alterations to swimming physiology, which can include reduction in the maximum swimming speed these fish can obtain, their ability to sustain high levels of swimming, and their burst speed. We also know that they can undergo respiratory distress, disruption to brain function, which can alter their natural behavior, and these can be expressed in their genes. Also, we know that hypoxia can cause direct mortality. So now I'd like to introduce you to my study species, which is rockfish, or the genus Sebastes. And this is composed of about 70 species that inhabit a wide range of habitats, from coastal nearshore systems to deep ocean systems that are low in oxygen. And so we see that there's this really broad range of oxygen tolerance that these fish already have. And these fish are, again, closely related. These rockfish are also an important recreational and commercial fishery, and they're part of the Pacific Coast Groundfish Fisheries Management Plan. So more specifically, I chose 2 species of juvenile rockfish to study. Both species reproduce through internal fertilization of eggs, where the mother then birth free swimming larvae. So getting into some of the differences between these 2 species, starting with copper rockfish, patrition of adults occur in the spring in the kelp forest, and then the larvae are then moved offshore for their pelagic duration, which lasts about 1 to 2 months. And it's generally thought that these fish might be found higher in the water column of about 0 to 50 meters, so potentially in that higher oxygenated water zone that I talked about earlier. During this time, the fish then transition into their juvenile phase before they recruit to the canopy, or the upper water column of the kelp forest, at around April to July. Blue rockfish, patrition occurs in the winter, also in the kelp forest. And these fish have a longer pelagic duration of about 3 to 6 months, where they are generally thought to be found a bit deeper in the water column. So maybe even in that oxygen water mass. They're potentially found in about 50 to 150 meters. That stratification zone can commonly be found at about 100 meter depth. So they can definitely be found in that low oxygen zone. They also then transition into their juvenile phase and then recruit back to the benthic area of the kelp forest habitat. So also in the kelp forest habitat, we have a little bit of a difference in oxygen saturation as you go through the water column, with higher oxygen at the surface and lower oxygen at the benthos. So getting in more specifically to what I wanted to investigate, I wanted to know if exposure to hypoxia would affect the exploratory behavior, the brain function, metabolic activity, respiratory function of these fish. And I also wanted to know, are there any species-specific responses? So are these 2 species of fish going to respond differently to the same oxygen exposure conditions? So the quest for these answers to these questions started in the coastal kelp forest, specifically Stillwater Cove in Carmel Bay, where myself and a team of divers would get in the water and target these juvenile phase fish that freshly recruit to the kelp forest, catch them with hand nets, and quickly transfer them into these flow trolls that we had with us underwater. And that's what a general catch would look like. After catching the fish, we then brought them back to Moss Landing here, where they were weighed, measured, and tagged using a subdermal elastomer dye tag. And this tag fluoresces under UV light and allows us to identify individuals as we want to choose them for each specific experiment. We often wanted to find a certain individual to put through an experiment. So this was a pretty stressful process. And so after the, forgot I had this laser pointer here. So this was a pretty stressful process. So the fish were given a recovery period before they were then transferred up to Santa Cruz at the NOAA lab, where we had our seawater manipulation system, where we were able to maintain 4 oxygen levels with a control at about 9 milligrams, which represent about 90–95% oxygen saturation, and then 3 decreasing oxygen levels with the extreme representing that hypoxia threshold. And so each one of these treatment conditions had 2 replicates. So we had 2 tanks for both the 2 the 4, 6, and control treatments. And these treatments were held pretty constant over the duration of the experiment. So here's some of the response variables that we investigated during this experiment, with 2 behavioral response variables, including escape response and behavioral lateralization, and the physiological ones, including aerobic scope, ventilation rate, and hypoxia tolerance. Now, it's okay if you don't know what I'm talking about right now. I just wanted to introduce you to them, and I'm going to get into each one specifically as we go on. So it did take a good amount of time to run through all of these experiments. It took about 24 weeks. So here's just a timetable showing the time it took for us to get through each one of these. So I'll start by talking about some of the behavioral responses, starting with the escape response. This is a fairly simple experiment aimed at testing for differences in behavior or boldness after these fish have been exposed to the oxygen conditions. So we created these 2-section chambers out of 6-inch PVC, where the top section was used as an acclimation chamber, and the bottom was the experimental area. So I have a little video here. There's a fish in the top there, which will become clear in a second. So the fish was given an acclimation period to get used to handling stress, and then that center divider was removed, and the time was recorded from when that divider was moved until when the fish leaves the chamber. And so we did this for all the fish across all the treatment conditions, and what we're looking for are some changes. So before I get into the results, I just first want to orient you to this figure. This is how all the figures to come are going to look. So on the left is the data for copper rockfish, colored in orange, and the data for blue rockfish is in blue on the right. On the x-axis is the treatment conditions, with decreasing oxygen going from right to left. So the control is on the right, and then the most extreme is on the left. And then on the y-axis is our response variable, which in this case is time. So now so we didn't see any statistically significant changes for this experiment, but if you take a look at the data for copper rockfish, there's this interesting pattern that emerges. And what that pattern is telling us is that there is this increase in time, the average time taken for these fish to find their way out of these chambers with exposure to low oxygen conditions. So if you're inclined to believe this, as I do, that this is a real trend, it gives us a little bit of an insight into what's happening with these fish. And so potentially what we're seeing are some behavioral changes in the brain function of these fish that are leading to changes in their problem-solving ability or exploratory behavior. Now, this is a long-term exposure experiment, and fish exposed to short-term conditions may respond a little bit differently. And the primary idea behind this is that fish exposed to short-term oxygen in their environment actually be able to move and seek out better oxygenated zones, actually escape from that low oxygen condition. But when given, when exposed to these conditions for extended period of time with no relief in sight, they may enact a different method for dealing with these conditions, and that would be a change in their overall energy budget or a reduction in their energy budget. And I'll get into that a little bit more as we progress through, but I just want to touch on some of the impacts, some of the effects that these changes might actually have in the environment, which are, so changes to their behavior may actually increase their risk of predation in the environment. So next I'll talk about behavioral lateralization. Now, behavioral lateralization is a measurement of brain functional asymmetry and reflects the bias for left or right turning preference, denoted by the dominant hemisphere of the brain. And this behavior is associated with cognitive tasks like schooling behavior, predator avoidance, and spatial orientation. So on the top right is a picture of the escape chamber, the behavioral lateralization chamber, which we measured this behavior in. And I know it's kind of hard to see, so I created this schematic here. And basically what we're looking at is this two-way T-maze. Fish was put into one side of these chambers and acclimated. And then it was then gently coaxed into this center channel and then down the channel where it was forced to make a turning decision, either left or right. And then this process was repeated about 10 times, and that gave us our results. And from that data, we were able to calculate the absolute lateralization index. This is a measurement of turning preference irrespective of specific direction and can tell us whether a population of fish are either highly lateralized or not lateralized. So looking at some of the results from here, we saw that exposure to oxygen conditions did decrease lateralization for copper rockfish. So again, this trend of copper rockfish being affected. And we saw our significant results in that moderately extreme condition of 4 milligrams of oxygen with a 60% reduction in lateralization. So lateralization is typically assumed to be advantageous for fish because it results in more efficient reaction time and aids in tasks that require directional response. So reductions in lateralization can translate to impaired cognitive ability, schooling behavior, spatial orientation, and escape reactivity. And the impairment of this behavior can make individuals or populations that are similarly affected, like copper rockfish, more susceptible to predation in their environment. Some of the causes of this can be some changes in brain function due to oxygen deprivation. And one of the possible things that are happening is changes in their neurotransmitter function or neuronal ion permeability as in a response to this low oxygen condition. So next I'm going to transition into the physiological responses, starting with the aerobic scope. Now aerobic scope is the capacity for aerobic activity of an individual, and it's the measurement of the maximum metabolic rate, the difference between the maximum metabolic rate and the minimum metabolic rate. So the metabolic rate of fish at their highest activity level and at their lowest. So to measure that metabolic rate, we use this instrument called an intermittent flow respirometer, which is pictured on the right. And now this is the experimental chamber section of the respirometer, and it's hooked up to an automated computer system. And so the 4 chambers pictured on the right, here's what a zoom in on one of those looked like. So a fish was put into a chamber, and it was kept in a closed system where there was an optical oxygen probe in line with the system, measuring oxygen. And so the only change of that system to oxygen is caused by the fish. So there was a computer system that was reading those measurements and running cycles, which allowed us to measure the metabolic rate of these fish. So for measuring maximum metabolic rate, we had to use a combination of a swim flume and the intermittent flow respirometer. So fish were put into this swim flume and swam at a high rate to induce exhaustion. And this was done in a standardized way that I'm not going to get into right now, but just know that it was standardized. The fish were then quickly transferred into the intermittent flow respirometer and ran to a point where we were able to measure their metabolic rate. So for the minimum metabolic rate, we measured this by putting the fish into these chambers here in the respirometer on the right and leaving the fish overnight so that they were at their lowest activity level. So in general, aerobic scope is a great proxy for fitness and can give us a little bit of insight into potential changes in growth and locomotion of these fish. So here's the results, and I'm first going to talk about this lower panel, which are the data for the minimum metabolic rate, which is also resting metabolic rate. And what we saw were no changes for any of the fish, for both species of fish across all treatment conditions. For maximum metabolic rate, this is where we start to see some changes in their metabolic activity, and the general pattern is decrease in metabolic rate with decreasing oxygen saturation for both species. Now these differences become real clear when we look at the difference between these two metrics in aerobic scope, with again some real strong decreases in aerobic activity, aerobic scope with decreased oxygen saturation. And we saw some pretty strong decreases, a 9% reduction for copper rockfish, and then a 64% reduction for blue rockfish. So these decreases in aerobic scope are primarily driven by that maximum metabolic rate. And so this is pretty interesting. What is this telling us? In a resting state, even though these fish are under extreme oxygen stress, they're still able to extract enough oxygen out of the environment to maintain metabolic function. And so oxygen is a key component in creating energy for these fish, Krebs cycle and electron transport chain. So we're seeing that they're still able to do that at low oxygen conditions when resting. But when they start to be active, that's when the differences start to come out. And we're seeing that they're not able to maintain normal function at these high activity levels under oxygen stress. So this inability to meet oxygen demands is attributed to their circulatory or ventilatory systems not able to move oxygen to the tissues that require it. And this can affect their fitness in the form of growth and locomotion. I just want to also point out that, again, there is a greater impact for copper rockfish in comparison to blue rockfish. So next, I'm going to talk about ventilation rate. This is a fun one. And so I created this chamber here. It's a 10 compartmentalized chamber where we can put 10 fish into this unit. And each chamber has an individual water source and water outflow. So this allows us to measure ventilation rate of these fish simultaneously, 10 of them simultaneously. So ventilation rate is a measure of breathing rate, analogous to breathing rate of any mammal, and is a measure of the opening and closing of the gill operculum. And so one opening and closing is one breath. So looking at some of the results from this, we saw that there was a pretty dramatic increase in ventilation rate for these fish exposed to low oxygen conditions. 50% increase for copper rockfish exposed to the lowest oxygen condition, and an 88% increase for blue rockfish. So now there's a minimum oxygen threshold that these fish are required to reach for normal function. And when put in oxygen stress, there's a couple ways that they can deal with that oxygen stress. They can change their activity levels by reducing the amount of energy that they're using, thereby conserving some of that energy for metabolic needs. They can also change their habitat, as I mentioned before, but we know that that option is not available to them in this experimental setting. And so there's this third option of an increase in ventilation rate, which is what we're seeing here. So the exposure to low oxygen can stimulate respiration, which increases their ventilation rate, thereby allowing these fish to move more water over the gills, increasing their extraction ability of oxygen out of the water. So transition into the next experiment, hypoxia tolerance. This is also a really cool experiment. And the explanation is a little tough, so I'm going to give an explanation, then run you through this figure here. So hypoxia tolerance is an experiment where fish are exposed to decreasing oxygen levels while their metabolic rate is measured. So as oxygen decreases, hypoxia tolerance, or their peak rate, is the point at which that metabolic rate of the fish changes from being independent of oxygen saturation to declining with further reductions in oxygen. And so what this looks like here is I have this figure here with oxygen on the x-axis. And so a fish would be exposed to this high oxygen concentration here, and then we have metabolic rate on the y-axis. So the fish, and this was done in this same chamber. We were able to reduce the oxygen exposure to these fish in a stepwise manner. And so as fish are exposed to a decreasing oxygen saturation level, their metabolic rate actually stays the same up until a certain point. After that point, their metabolic rate begins to decrease with further reduction of oxygen. So if you put a regression line over these two sets of data, the area where they affect is their peak rate value or their hypoxia tolerance. So here's the results from this experiment, and what we saw were that both fish had an increase in hypoxia tolerance. 16% and 14% for the lowest oxygen exposure conditions for copper rockfish, and a 33% increase in hypoxia tolerance for blue rockfish. So this increase in hypoxia tolerance with declining oxygen exposure for both species tells us that these fish actually are acclimating to these conditions to some degree. And so there's a few methods that these fish may be using to acclimate to these conditions, and one of those is an increase in ventilation rate. And this is what we saw. If we go back to our ventilation rate data here, we saw that there was actually an increase in ventilation for fish exposed to those low oxygen conditions. This is potentially allowing these fish, this is actually like potentially that acclimation ability of these fish to these low oxygen conditions. But there's also a couple other processes that these fish may be using to acclimate to these conditions. And those allow them to increase the extraction of oxygen out of the system. And those include an increase in blood flow within their bodies, or an increase in hemoglobin content, as well as the binding between hemoglobin and oxygen. So hemoglobin is that main receptor in the blood that binds to oxygen and moves it throughout the body. So an increase in those would allow a better extraction of oxygen for these fish. Additionally, there's been studies that have showed that some fish have the ability to actually adaptively remodel their gill structure. And what that would look like is under oxygen exposure, we have fish exposed to normal conditions and then progression of hypoxia exposure up to 7 days. And what we're looking at is a change in the secondary structure of the gills. And so essentially what's happening here is there's an increase in surface area of the gills, which is allowing these fish to better extract oxygen out of the environment. So one or more of these processes can actually be occurring. And this can be occurring in rockfish. We don't quite know yet if this is occurring, but this is actually something that students are currently looking into for rockfish in response to oxygen. So if you want to know whether that's actually happening, you're going to have to come back and check out one of the other ichthyology lab thesis presentations. So that's all 5 of the experiments that I ran. So now I want to kind of summarize some of the results and give you a little bit of insight into what they mean. So getting back to this question of how will juvenile stage rockfish fare under future oxygen conditions? Well, we saw that there was a decrease in aerobic scope. So generally we saw that there were these impacts to physiology, to a lesser extent behavior. But we saw a decrease in aerobic scope, an increase in their hypoxia tolerance, and an increase in their ventilation rate. So some of the implications of this are, starting with behavior. So behavioral changes can indicate that there are some changes occurring in the normal brain function of these fish, which can alter the way that they interact with their environment. Deviations from normal behavior can actually increase their mortality rate in the environment and make them more susceptible to predation by higher trophic level predators. So stepping back to those first 2 experiments I showed you, we saw that there were changes in the exploratory behavior of these fish for copper rockfish and also their lateralization. Now lateralization in particular is particularly associated with escape reactivity. So a highly lateralized fish is actually better able to escape a predation event than a less lateralized fish. So if these changes are occurring, we might see more mortality. Now for behavior, as I mentioned before, oxygen is a really important component of creating energy for these fish, potentially as important as a food source. And it's because of its importance in these key metabolic pathways that create energy, that being the Krebs cycle and the electron transport chain. And reductions in these processes through oxygen stress can actually shift the energy budget of these fish, force them to reallocate energy from non-essential activities like growth and locomotion to essential activities like metabolic function and keeping them alive in the present. So these decreases can also lead to some increases in mortality in early life stages. So we can see that these fish are not able to swim to their normal capacity, which can then lead to increased predation events. So we asked the question of whether these species were going to experience any responses different from one another. And what we saw is that copper rockfish were generally more affected by exposure to low oxygen conditions. We saw that copper rockfish had a decrease in lateralization, this trend of increase in escape time, and an increase in ventilation rate, while not as strong as that of blue rockfish. When you pair that with their only relatively minor increase in hypoxia tolerance, we can see that these fish are working really hard to try to extract oxygen out of the water, but not doing it as efficiently as blue rockfish. And so what we're seeing is that copper rockfish in general are more sensitive to oxygen fluctuations than some other species that are closely related to them, like blue rockfish. So from here, we ask the question, why are these differences occurring? And what I think is happening is that it's related to their life history characteristics. Specifically, during their early life stages. So if you'll recall from the beginning of the talk, I talked about how these two species have different early life history characteristics where they're born at different times and then they have different offshore pelagic durations where the copper rockfish could be found up in the upper water column and the blue rockfish in the lower column. So potentially there's a difference in exposure history here. Blue rockfish in higher oxygenated waters copper rockfish in higher oxygenated waters, blue rockfish in lower oxygen waters. So there may be this pre-adaptive ability of blue rockfish who are exposed to low oxygen at early life stages so that they're actually better able to adapt to deal with these conditions later in life. Now also these adaptations can occur and do occur over a generational time period. And so this process is occurring every generation. And so little by little, there could be these changes in the genes that are passed down from one generation to the next, which are allowing some species to better adapt to low oxygen conditions. In general, a little bit, kind of later in their life histories, in general, blue rockfish have a higher activity level, higher baseline activity. They're usually found up in the water column swimming around. And so when exposed to oxygen stress at high levels, they also might be better able to deal with these conditions. And what this leads to is winners and losers, even between these closely related species. Supposed to do that slide before. So I can't end this talk without talking about the occurrence of multiple stressors. As I mentioned in the beginning of the talk, this talk is focused on the idea of upwelling here in Central California. And what occurs with upwelling is both the occurrence of low oxygen as well as pH and low temperature. Here's a figure showing pretty representative conditions of these three metrics, or these three stressors during upwelling events. And what we can see is that all three are actually changing together. So when these fish are exposed to low oxygen conditions, they're also exposed to low pH and low temperature. So this can have some additional effects on these fish, potentially interactive or cumulative effects. So I just want to give you one example of some studies between my research looking at exposure to oxygen and a previous student, Will Fenney, who looked at exposure to pH. And so at the top, this is data that I collected looking at aerobic scope, which is the difference between the maximum and minimum metabolic rate. And what we saw was a decreasing trend of aerobic scope with exposure to low oxygen conditions. Now below here, this is the same experiment repeated with exposure to differences in pH, with the lowest pH or most extreme conditions on the left side. And what we're seeing is the same general trend for copper rockfish of decreasing aerobic scope with exposure to more extreme pH conditions. So because these fish are being affected in this way when exposed to these conditions individually, we might expect that there could be a cumulative effect when exposed to these two stressors together. And again, this is some work that's actually ongoing in our lab. But also, if we look at the effects, the responses from blue rockfish, we saw that blue rockfish had this same negative response of decrease in aerobic scope with decreased oxygen. But when exposed to pH, no change. And so this is another example of differences between species and how they interact with these conditions. Now also, these fish are in a constantly changing environment, and they're going to be having to deal with the consequences of climate change. And so some of those consequences include an increase of upwelling events leading to stronger and more frequent durations of low oxygen, low pH exposure. So these fish are ecologically and economically important. They're a key prey item for a number of higher trophic level predators, and their early life stages are predicted to be most critical to changes in water chemistry, like changes in oxygen. These early life stages are also most critical for population replenishment, so impacts at these stages have some pretty large effects for adult populations and recruitment success. Going forward, it's important to investigate the impacts of multiple stressors on these species and to expand to more species to really get a broader idea of how upwelling and climate change will affect a wide variety of species with differing life history characteristics. So that's all I have for you today. This work would not have been possible without a lot of people as represented by all these names up here. And so I just want to thank a couple people, including my collaborators, as well as my committee members, who are all instrumental in pulling off this research. We had a lot of research support, including a whole slew of lab assistants from students and interns who helped maintain these fish and run these experiments. They were an integral part of this research. Also, my ichthyology lab mates were really supportive over this whole process and often helped catch fish or run experiments or look over talks, etc., really adding to this whole process. Of course, I'd like to thank our facilities guys who have really been great. As you saw from Scott's talk, I'm a bit of a tinkerer, and they really welcomed me into their area, into their shop, and let me play with all their tools, which was really great. Also, I'd like to thank the Marine Ops crew. You guys have really made this last year here or so, year and a half, a really great experience. You're all awesome people to work with. Also, thanks to the IT team who actually have set this presentation up today, allowing it to be streamed online. So we have another audience online watching, at least my parents, hopefully more than that. And now I think you'll notice that there's one name missing from this list. And so I want to give a special thanks to Scott Hamilton, who in addition to being an excellent advisor, is also the party. And I'm sure he's going to be celebrating like this sometime later tonight, knowing that he successfully graduated another student out of the lab. And so with that, I'd like to thank you for all being here, and I'm going to open the floor for any questions that you might have. Any questions? Christian.
So you kind of talked at the end about how the copper and the blue rockfish being at the surface down at the benthos of the kelp forest have these different responses to all these different tests. I'm curious whether you have any idea, though, of how the surface waters versus the deeper waters in the kelp forest respond to increasing amounts of upwelling. I could imagine that as you get increasing amounts of upwelling with more and more hypoxic waters or low oxygen waters, that the surface waters might respond as far as their oxygen saturation. They might have less of a response to that because they're mixing with the air and they're being oxygenated. You know,
Yeah.
however much low oxygen water comes up, they get oxygenated.
Yeah, so
do you expect there to be kind of differential responses between surface and deeper waters as you get increasing amounts of upwelling?
Yeah, I think to a certain extent we would. Now these currents are kind of constantly going, so it's moving these water masses in an ongoing fashion. But the surface waters, there's a couple things that are happening here. So you've got these algal blooms at the surface, and so there's these primary producers are actually adding oxygen to the system, as well as there's wave activity, which can also add oxygen to the system. And then as they die, they're then sinking down into the water column. And really, that's where I think the biggest difference is that there's that microbial decomposition occurring and the associated respiration at depth. So I think that the lower water column is actually going to decrease at a faster rate than the upper water column. So yeah, potentially these copper rockfish, which recruit to the canopy, may actually be a little bit, have a little bit of a buffer from that. Again, as these fish progress through their life. They change their habitat, but yeah, generally I think that's what's happening.
I had a couple questions, and I think I missed the first part. How did you manipulate the oxygen level?
Oh yeah, that's a good question. Yeah, I'm sorry I didn't touch on it, but yeah. So in our seawater system, we had a series of water tanks, and so we had one big water tank which flowed to a series of 4 smaller ones. And so there's a constant flow through of these waters, and then those 4 fed into our experimental tanks. But in those mid-sized 4 tanks, we had like air stones that were bubbling nitrogen gas into the water. So the nitrogen gas was actually removing oxygen out of the water. So we had oxygen sensors that were put into that water, and they, through a solenoid, they turned the gas on and off to maintain that level where we wanted it.
Did you happen to have pH sensors in the same?
Yeah, so there's a whole other component of this research that I didn't talk about, but we pretty much did the exact same thing that I just talked about with you all here in relation to oxygen. We replicated that for pH. So this project was actually twice as big as I just presented to you today. So the whole thing was repeated for pH, again with pH sensors. And for pH, we used CO2 gas bubbled into the water to manipulate the pH level.
Okay. Then another question, is anyone looking at the feeding behavior of the juveniles under these?
So that's not something we actually looked at, although I think it is actually really an important question to ask, because exposure to low oxygen conditions can change their feeding behavior. Just by watching these fish, we did see some changes in feeding behavior where we fed a standardized amount of food to all of the tanks, and the following morning we would go back and clean out any excess food that was left at the bottom of the tank. And what we were noticing is that there was actually a little bit more, actually a significant amount more food left over in these low oxygen tanks compared to the control. So I think from just watching these fish, we can say that there was a decrease in feeding in the low oxygen conditions as well.
Yeah, you could actually close the energy budget if you have all that information too, right?
Yeah.
You know, the caloric content of the food and.
Yeah. Thanks for those questions. Genji?
So it's interesting to see these different responses based on different early life histories, and I want to know if you know if anyone else is doing studies on other species. So if you compare those responses of all juvenile pelagic rockfishes versus all
Yeah, I think that's a big question, and our lab is actually, as well as in combination with Cheryl Logan's lab at CSUMB, who they're kind of spearheading the gene expression part of this project. We're trying to expand this research to look at more species. So we've included gopher rockfish, and are there any other species, just gopher rockfish? But also, this experiment is also being done up north in Humboldt area, where they're looking at effects of the same conditions with some other species as well, and they've got black rockfish up there. So there's also a geographical component of this. So yeah, we're trying to expand, but as you saw, this work takes a really long time. Just this experiment took 6 months, and it's been ongoing for years, and we've kind of just covered about 3 species so far. So yeah, it takes a long time, but definitely we're working at it.
Are there current preliminary results for the black rockfish?
There are, but I don't.
They represent.
Yeah, I don't know what they are. But they are. Yeah, Jake and Kristen, Melissa will fill you in. Anything else?
Quick one. Are fish aware enough to where they can say to themselves, all right, it's getting really hard to breathe around here. I'm going to go somewhere else where it's easier to breathe. And if that is true, then could this lead to potential shifts in geographic distribution of species?
Yeah, I think the answer to that question is yes. And it's been seen in their environment that fish actually seek out better oxygenated areas. So yeah, that can lead to changes in the distribution of these species, potentially over large geographical areas. Yeah. Right on. Anything else?
I'll ask one. Right. So you did these fairly long-term experiments, right, over a
period of months, these chronic exposure to low oxygen. So if you think if you did the same tests, but you expose the fish just for a couple days, kind of a normal upwelling cycle, you'd see the same responses for all of them, or you think?
Yeah, I think generally we would see pretty similar responses. I think these fish are able to bounce back after if they're exposed to short-term conditions. I think that they're able to bounce back a little bit quicker, but generally, I think that we would see these responses.
Anything else? All right, so we're going to take Evan into the back room and interrogate him a little bit more, and you guys can start the celebration. Yeah, thanks.